Okay, I think we'll make a start. Welcome everyone to our Ecological Continuity Trust webinar series. Today we have three speakers. First of all, we're going to hear from Mara Lawniczak and Lyndall Pereira, who are from the Sanger Institute. And they're going to talk to us about BIOSCAN, which is a flying insect diversity by DNA sequencing. And then we're going to hear from Kris Hart, who's from the Birmingham Institute of Forest Research, who's going to talk about his experiences of working with BIOSCAN. Just a couple of housekeeping reminders, so please keep your microphone muted and your video off during the presentations. And if you have any questions, please pop those into chat as they occur to you, and then at the end of the presentations, Ben will go through those questions and invite you to talk about them. So thank you very much, and over to Mara and Lyndall.
Thank you so much, Carly, for introducing us and for having us here today. We're really, really excited to talk to all of you that are part of the ECT. And so I'm going to tell you a little bit of the kind of high level view of what we're trying to do and why and who we are. And then Lyndall will tell you all of the details of how it works and what you would gain from it. So we work at the Wellcome Sanger Institute, which actually, if you've ever heard of it, you probably think, what are we doing with biodiversity? Because it was very involved in sequencing the human genome, the original human genome. But in the last few years, we've gotten very involved in trying to contribute to the global efforts to understand biodiversity from a genomic perspective more deeply. And so we formed a new department, effectively. We call it a program, the Tree of Life. And we have faculty members in this unit. There are 4 of us now, so we're growing. And we have a team of around probably 80 to 100 people now who are all working in this space to try to use genomics and genetics and sequencing to improve our understanding of biodiversity. We have 2 flagship projects. So down in the bottom, the blue, can I shoot? Yeah, you can probably see my mouse. So this is our logo for the Darwin Tree of Life project. And then this is the project we'll be talking about today. The Darwin Tree of Life project and BIOSCAN, I think, are quite complementary. And they're basically Sanger's contributions to global endeavors. And those global, oops. Right here? Okay, sorry, not my computer. There's these global projects towards which we're contributing are the Earth Biogenome Project. And the vision of this project is to create a high quality reference genome, which means all the A's, T's, and G's that make up an organism's genome for every described species on the planet. And then on the right, you see the International Barcode of Life, which has launched a new program of work called BIOSCAN. And that's the vision that we're contributing to. BIOSCAN is a kind of ramped up effort to really do high throughput species discovery. So globally, the ambition is to create DNA barcodes for 10 million more specimens. And then of interest to us is the space in the middle, which is the species interactions, which Linda will talk a little bit about. We haven't actually made progress in this area in a deep way yet. So currently we're still really doing species discovery and trying to create a barcode for every specimen. But what we're really trying to get at is to develop methods to actually understand, using DNA, who these individual arthropods have interacted with. What are they infected by? What are they pollinating? What are they eating, etc. So she'll talk a little bit about that. Okay, so, and the Earth Biogenome Project, which I won't talk about much today, is this effort to sequence the reference genomes. So in summary, these global projects, Earth Biogenome and BIOSCAN, are happening all over the place. Lots of people are contributing to them. And in the UK, Sanger's contributions are these 2 flagship projects, the Darwin Tree of Life and BIOSCAN. So briefly on the Darwin Tree of Life, it is also a highly collaborative project across the country with lots of biodiversity experts involved, going around identifying, collecting, and contributing specimens to get reference genome sequenced. We are this is a really big project with hundreds of people involved now. And we have lots of different interfaces with the data. So you can go and understand which species are actually already collected at this portal. And you can understand where in the process of getting a genome sequence are they. So over here, for example, you can see this is an actual genome note at the end, which is our final product. So we basically, for this particular butterfly or moth, I'm not sure, we have extracted this DNA, sequenced it, assembled it, and put all the information about that, as well as the sequence up on the databases, and finally written a little note to tell you about that. So now I'm going to switch completely to BIOSCAN, but I wanted to give you that view on Darwin Tree of Life because BIOSCAN complements Darwin in our future vision of how BIOSCAN will work. So BIOSCAN is really aiming to have 100 sites across the country operating just for a very short period, so 24 hours every month, but for multiple years. So we're getting this kind of longitudinal and spatial sampling of what is flying around. And we use the Malaise trap. We recommend using a Malaise trap because it's a very passive way of collecting a lot of organisms at the same time. There is an opportunity if you have other interests in other types of traps that you're doing, other types of collections you're doing of arthropods to feed them into this project. But our main drive is to try to be sampling in consistent ways across the country for consistent periods of time so that we can really start to piece together how do these organisms change over space and time? What's there and how does it change? And that can be just because of sort of normal variations through the year, but it can also be because, for example, some of our partners are putting into place rewilding activities. And so really getting that early upfront understanding of what's there and trying to measure over the course of the years that we can run, how is that improving? Is it working to improve the diversity of what's being sampled there? So we send you a Malaise trap for your site, and then you run that, you get all the consumables. This over here is called a plate, and it's got 96 little wells in it, little dishes. And what we send you those pre-filled with liquid, and you then take the individual organisms out of the trap and you plate them in, and then you seal that plate. And then you record metadata associated with that, so when, where, how. And then you give all of that information back to us and all of those samples back to us. And then we do a non-destructive extraction. So this is kind of a view on that. These are little individual insects in those wells, zoomed in, and they're leaking their DNA into the liquid. So the carcass remains intact, and then you can have those back so that when you get the DNA barcode back, you know what it's been identified as. You have something to match up with it if you want it. Some partners really want that, and some partners actually are not so interested in getting that back. They'd rather it go to a museum. So in that case, we would send it back to a museum for you. Then we take that DNA and we just use a tiny little bit of it to sequence the DNA barcode, which tells us what that specimen is. We present you with a biodiversity report card, which Linda will do a demo of, and that will tell you how your site compares to last month or the previous years, that month, or how it compares to other sites in the area or across the country. So our ambition is to explore 1 million arthropods by the end of our project. And a big part of this project is to create this DNA biobank. So we're just using a tiny little bit of DNA, trying to understand what's out there. But that DNA is of major value to research and biodiversity studies in the future. And so all the rest of that DNA we retain, and maybe we will use it for some other projects like the Darwin Tree of Life that I talked to you about, or maybe it will be used for other studies, and it may end up, most of it will probably end up in a repository like the Natural History Museum or National Museum Scotland. I'm never gonna get this right. Okay, this is all done very high throughput. So we're looking at over 9,000 specimens every time we do what we call a sequencing run. So we need to get 100 of those plates in, extract the DNA, run the PCRs, combine them all, and run them on a PacBio machine to get those molecules sequenced. So everything is done with robots once it gets here. And this is kind of part of how we have this partnership with you, with others around. We can't possibly get enough people out there collecting at this scale, but we can do this DNA work at this scale. So if we have lots of partners who are willing to collect because they get the information they need back, we can actually scale up the sequencing endeavor at a level that really is not possible to do in a normal lab. This is the machine that everything gets sequenced on. It's called the PacBioSequel 2. And it's a kind of incredible machine. Actually, the DNA just goes, this is only about a square inch, this little chip. And it goes into this machine and the DNA gets loaded here. And basically, cameras are recording the A's, C's, T's, and G's to tell us what each individual specimen is. Okay, so we have finished the first, actually a bit more than a year now of our pilot. And all the partners down on the bottom have been working with us to get the whole system kind of pieced together and up and running pretty smoothly now. These are a couple of actual malaise traps out at actual sites. And we are so grateful to our early partners who really worked closely with us to get the whole thing operational. So I think this is nearly my last slide, and then I'm going to hand over to Lyndall. I think I've tried to convey to you that these BIOSCAN and EBP are these kind of global visions to catalog through barcoding or reference genomes all of Earth's biodiversity. In the UK, we have 2 flagship projects that are contributing to those global visions, and those are BIOSCAN and DTOL, Darwin Tree of Life. In my group, so I'm just a scientist, I'm a faculty member at the Sanger Institute, we run BIOSCAN, but we're very involved in Darwin Tree of Life, which is, as I said, a very collaborative project across the country. So in my group, we're aiming to catalog insect biodiversity across the country and give back to our partners all this kind of what we think of as immediately useful information through these biodiversity report cards you'll see a demo of in a minute. We really want to create these future-proof DNA biobanks at scale. So by the end of the project, we really hope to have 1 million DNAs ready to go for other work. And we're going to start to explore a very tiny proportion of those DNAs. So for example, in dipteran families where there's a lot of species hyperdiversity, we might start to do additional sequencing on some of those individuals. But obviously we don't have the ability to sequence 1 million genomes. We have the ability to sequence 1,658,000 bases, but that is a very different scale of sequencing. And this is my final slide. So we think that BIOSCAN will reveal new species in the UK, and it will also help us monitor how species diversity changes over space and time. We really think that the future of biodiversity monitoring is DNA based. It can scale things up massively. There's all sorts of exciting stuff happening around the world in this area. For example, really cool work actually capturing air to understand what DNA is floating around, to see who's actually in that area. So the environmental DNA concept is just getting bigger and bigger. And without these barcodes to really understand how each, what is each of these things we're sequencing, we actually, the environmental DNA work is less kind of informative. So this is a very good foundation project to understand what these barcodes really connect with. We're focusing particularly on insects, in part because I've always worked on insects, but they're also critical to the health of our planet, and we don't know what we're losing. Everybody knows about the windshield stories where you're driving and you're not seeing the kind of insects splattered on your windshields that you used to, and we are really losing insects at a vast rate. So we want to usher in this new era of DNA-based biodiversity monitoring. And we believe that these resources will help us understand the impact of climate change and of conservation efforts on how we are trying to protect our biodiversity in insects. And with that, I will end. And thank you for your time. Linda will shift over to her presentation now.
Thanks, Mara. All right, so I'm going to dive into some of the details of BIOSCAN. And to start off with what you can expect from us, and then a bit of what we're asking from you for the project if you decide to join. So we provide you with the malaise trap and the collection kits, enough preservative for each collection event for the duration of your project, labeled 96 well plates with the preservative in ready for specimens, a standardized manifest with careful metadata collection, all the forceps, capping and decapping tools, sorting trays, and any other consumables, the shipping materials, as well as shipping costs if required. And then when the samples come to us, all the lab prep work that's done for the DNA extraction, all the sequencing costs for all of your specimens, and then open access to all your and all other BIOSCAN data, so that's raw data, as well as the biodiversity report cards. And then we also send your specimens either back to you or deposit them in a museum or repository of your choice. So what we're asking from you is that you have the collection permission for your site of choice. And then there's the staff and volunteer time for trapping and plating. So this is dependent on how long it takes for you to get to your site, the season that it is, and the number of people involved. So usually for just one person, it takes about an hour to set up and remove a trap, and then about 2 to 6 hours for plating and metadata capture. So, and this plating time is reduced when there are more people who are contributing. Then there's the to have an adequate lab space for sorting and plating insects, then travel costs where relevant, and shipping costs where possible. So what do we require from our partners? So currently we have 24 active partners. We have a map on the side, 34 traps that are active, and then 8 traps pending. So that's initial conversations and onboarding still to happen. So we're launching, as Mara mentioned, we're sort of launching this project into like full scale now. We're looking for 100 sites. We want to get 10,000 specimens coming in per month. So our top requirement for now is that you can start now. So we don't have restrictions on our site location and habitat for now, but this may change in time as we do start onboarding more partners and sites. So when looking at sort of the sequence analysis and repositories, so the data that I mentioned that you'll have access to, we upload the raw data file to this online platform called mBrave, and it's linked to and connected to the Barcode of Life database. So, and then we run the parameters, and then there's some quality checks that we do. So we can provide you with the raw data so that you can do your own pipeline if that's what you're interested in, or the demultiplexed data we make available as well. And that demultiplexed data is the data where it's been matched to the reference database, and so you get sort of a list of species names and sequences. So that is then deposited on the Barcode of Life database, or BOLD as we call it, and that will come up, so you'd be able to search for it. It's open access. It'll come up in a list that you can see down here, and then each specimen that we sequence will end up with its own little page with a photo and all the metadata that you collected, as well as the sequence data. And then an extra thing that we're doing is that we're sort of collaborating with the NBN Atlas, and all records that we collect will also be deposited into their database. So onto biodiversity report cards. This is sort of the setup that we have. We use Google Data Studio, which is a free software. And what I'm going to show you now is a demo, and it's with mock data, or mock metadata. So just a brief cover of sort of the barcode index numbers we've called bins, if you've heard of them before, or OTUs, which is an operational taxonomic unit. There's a lot of that sort of features in these biodiversity report cards, and the idea of the bin is that it refers to these clusters of similar DNA sequences, which often reflect the actual species, but it can also distinguish between populations of the same species. And so we also have a look at the scientific names and the species that we have collected, but then not all the species in the UK are in bold. So we sort of present you with both. We present you with all the bin numbers. So if the species isn't on bold, you have a record of it with an actual identifier. And this biodiversity report card also has a couple of pages on the side here. You can see map of sites, sunburst, etc. And I'll go over those now. So this is the moment where I try and play a video, and hopefully it works. So this is just a screen record, and I'm going to go over it. So this is the map of all the different sites. You can see the sites at the bottom. We've got filters, so you can filter by partner, whichever catch, month, or even the order of the insects. So here I'm selecting a number of partners that I might be interested in, sort of comparing what was found. And so it shows you with the different bubble sizes, what's been collected. If I'm just interested in, say, July, it shows you the bubble numbers change. And they say I'm just interested in looking at the wasps, so just click on that. And then that should change the size of the bubble. You can also go and hover above the bubble, and it gives you a little bit of extra information. And if you want, you can export it as a CSV and do for reporting purposes or for further analysis. You can reset back to normal. Then we have a sunburst chart. So this is a chart where it shows you proportions found or sequence in that run or whatever filter you use. You've got the order level, and then we've got the family level, genus, and then right at the end, we've got all the different species, scientific names. As I mentioned, we've got the usual filters at the top. So in this case, we just want to have a look at, say, our university, and you can choose a month if you like, and you can see the proportions change. So you can also click through this actual plot. So if I want to have a look at the mustards, you can click on that, and then that will bring you all the way down to the mustards, so you can see the different genera and the species. If you want to go back, you just click in the middle, and then it takes you back. So you can interactively click through the taxonomy for what you're interested in. And then we have this comparison plot or comparison bar chart with a pivot table. They are linked. We have a bunch of filters at the top. So this is the pivot table that shows you your July and August. So it kind of gives you this time series or timeline of what you've collected. And you can see when it was present in July or August, or you can see their presence and absence. So if we select the partner that we're interested in, we can also filter by order or family. We can also click on the bar chart itself. So you can do 1 month, or click the other one, do 2 months. So here are all the Coleoptera found across the months. And then the pivot table showing how many specimens were caught, and export the data again if you want. Let's say you're interested in maybe a particular family. So you can actually just type that in, as long as you get the spelling right. And that will just bring up a list comparing the 2 months that you can export as well. We also provide you with a full list for export. So that's the sort of different codes and IDs, the scientific name or the whole taxonomy that it matched to, including the bin number or OTU number that has been assigned to it. There are 6,000 in this list, so I'm just going to filter it by partner, which can sometimes take a little bit of time. And say we're just interested in one of the months, and then we want to maybe export this for some further analysis that we have a look at, or some sort of reporting. So we've also included a sort of conservation status aspect to this report card. So whatever was found in the sequencing, we match it to the JNCC designations. So if we choose our partner that we're interested in, and maybe our month of collection, it gives you a list of everything that was found in that month for that partner in their malaise traps. And say you're interested only in any sort of conserved Diptera, we click on that, and then it brings it up. It also gives you a whole status description, so you can actually go and look up some literature if you want. Then we have this compare lists version, so it gives you a bit more freedom if there's sort of 2 sites you want to compare, or 2 aspects that you'd like to compare. So in this case, we'll compare the 2 universities, so University 1 and University 2. And you can also click on this list. So if we're interested in Hemiptera, we'll click on these 2, and we can actually drill down. So we click on the little drill down button, and now we can see how many specimens were found, or I mean, how many scientific names are found with each family. We can drill down further, and now we can have a look at exactly what's been found species-wise. And again, all exportable if that's what you wish. And then finally, we have this species index information page, which is from Pantheon. So this links to that database. So there's this potential to link to other databases. Here we've got, so there's SQS score, as well as some ecological data that's associated with the names that are found in the traps. So if we do the usual where we choose the partner that we're interested in and maybe the month, then we can have a look and see what was found, and we have a little bit more information about it. So hopefully adds a bit of value to that list of species that you get. Okay, that's my demo. There we go. So Mara touched on this a bit earlier, but this is just a bit more detail on those sort of future plans to analyze the DNA for what we call mini barcodes to monitor the ecosystems. So, and this is one of the reasons why the DNA and biobank that Mara mentioned is just so important, because while we're not at this stage yet, we're making good progress to get there. And it will take some time, but when we're there, we'll be able to use this DNA that we have in this biobank and be able to look at each one of the specimens for exactly this, the cobionts, the parasitoids, any other groups that are associated with it, and then sort of, build and then hopefully monitor these ecosystems in the future, and sort of see patterns across space and time of how these things are connected to each other across the different species. So some summary highlights, basically start collecting now, register your interest today. I'll tell you how to do that in a moment. And well, insects are great for biomonitoring. You don't have to be an entomologist to do bioscan. There's an opportunity to learn insect ID skills if you desire that. You can create a baseline for your site. You have this opportunity to connect to other data, other sites and other institutions. There's helping build the UK Barcode of Life database of reference species, so that, as Mara mentioned, that sort of makes the eDNA side of monitoring and barcoding it's a bit more informative. And then, of course, your voucher specimens are not destroyed and they're deposited in museums. So they're available for future reference and for any taxonomic work. So yeah, this is how you can register your interest. So we have a web page. I've got the link over there, and there's a giant orange button that you can't miss. So just click on that, register your interest, and then I will be in touch. Thank you.
I guess that's over to me. So hello everyone, my name is Kris Hart. I'm the operations manager for the Birmingham Institute of Forest Research BIFoR-FACE facility, which is in a mature oak woodland. And we're one of those little dots that was right in, well, one of the only dots that was in the middle of England. And I don't have a presentation, so all you've got is my face to look at and my wispy voice because I'm starting to lose it. I really just wanted to have a couple of moments just act really as a testimonial. So our institute used to very vigorously monitor the insect populations while we had a PhD student here who's principally focused on entomology. And when he graduated, it kind of all fell away, even though as an institute, we all absolutely recognize the importance of entomology and the biomonitoring. It would appear that there wasn't much funding or support from the university for that aspect. So internally, we continue to monitor, we continue to put our malaise traps in place, pan traps, pitfall traps, and various other monitoring methods. And it's kind of becoming inactive. So when this opportunity came along, is a real, a gift horse to us. And we've embraced it with both hands. And it really, really did seem, just again as a witness testimonial, too good to be true. And until I met Lyndall, and she very kindly came and visited the site and demonstrated a lot of what she just said, the professionalism and the expertise behind all this is just phenomenal. I've never known a support package like it. And I really wanted to take the opportunity just to communicate to this group that this is a really, really great opportunity in order to build our network. And we believe, from my institute's point of view, from the University of Birmingham and BIFoR, that the data that we will get back from this is going to enable us to leverage funding in order to reinvigorate some life back into our entomological research field. And we just supremely see the value, to the point where, based upon everything what Lyndall and Mara have just expertly demonstrated, I was able to take some of these key messages back to the university and leverage direct funding immediately in order to process all of our archive. So one of the things that we've been very lucky to do, thanks to that previous PhD student, Liam Crowley, who I think is out there somewhere as one of the guests, but we are able to go back and now get all of our samples sequenced from the last 5 years and for the next 5 years. So eventually we'll be able to build a 10-year-long database, giving us species in abundance. And I could be wrong, and I'm happy to be wrong, but I'm pretty sure that that kind of a database will be unprecedented in the UK. So at least that level of detail. So I can see the power in this, and I hope you guys can as well. And that's really all I wanted to say. So I'll hand back to you, Carly.